Welcome back, everyone. We have reached to the final session of the day. In this session, our esteemed panelists will discuss in depth genomics, genetics, gene editing, and bioinformatics. Very interesting topics. To start, it's my pleasure to welcome the chairman of the session, Dr. Paolo Martini, Senior Vice President of Moderna. Welcome, Dr. Paolo. It's great to have you with us at the opening of the last day and even the closing session of the day of the Riyadh Global Medical Biotechnology Summit. The mic to you.
Thank you so much. It's a pleasure, it's an honor for me to be part of this meeting. So thank you again for inviting me and to be part of this incredible endeavor that you guys put together. It's incredible. I'm chairing session 9 together with I would like to call my friend and colleague, Dr. Majid Al-Fadel. We know each other for a long time right now. And I'm so proud to be able to introduce him today in this session about genomic genetics, gene editing, and bioinformatics. He's going to give us a lecture on genetic and rare diseases programs, GARD, an opportunity for an orphan disease biotechnology. So I'll give it up to you, Majid. It's my great pleasure to introduce you. Thank you.
Thank you, Paolo, for this nice introduction, and thank you for the scientific committee and organizing committee for inviting me to present in this important summit. I will talk today about genetic and rare disease program, which we established in our research center in Kmark, and what is the objectives and what is the achievement so far, and what is the future plan. So rare genetic disorders are individually rare, but collectively common. We are 10 times higher incidence of this disorder compared to USA and 4 times higher than Europe. Discovery in rare genetic disorder lead to discovery in complex genetic disorder. So as you know, we have one body, so any discovery in genetic rare, any discovery in a single gene defect with this lead will open a horizon to discover in complex genetic disorder, and we have a lot of examples in the literature. For example, for obesity, congenital leptin deficiency is a single gene defect, while obesity is a complex genetic disorder. So the scientists now, they are working to find a treatment for obesity through this single gene defect. Another example is mitochondrial disorder. The only, or one of the barrier for establishment of genetic and rare disease program was the high cost regarding the medication and the treatment. Why we are care about genetic and rare disease program in Saudi Arabia? Because we have a 3 sites in Kmark. It is distributed all over the kingdom. We have over 500 staff. We have around 50 scientists. And in Kmark, we have running 200 projects. And also we have one of the biggest biobank in the Middle East, which expected to collect 200 samples, and infrastructure is available. From the genetic point of view, we have 5 genetic consultants, 2 assistant consultants, and really well-equipped staff to do such program. On the other hand, one of the rationale, there is a huge expansion of the genetic disorder over the years. So this is from statistic. As you can see, actually this is 2 years ago. 2 years ago it was 4,274, and yesterday I checked statistic. We reach around 6,000 diseases, which is phenotype known and molecularly known. And this indicates that we discover around 1,200 new diseases into years. And as you know, we have around 30,000 genes discovered, and only we link to human around 6,000 so far. So still there is opportunity to discover more and more until we know the capacity of genetic disorders. Again, other rationale that you know now, actually the phenotype become more related to the genotype. So you could see now 2 or 3 phenotype related to a single gene. Before, only we have 1 phenotype and 1 genotype. Currently, you can see that the number of gene with 2 phenotype and 3 phenotype and 4 phenotype is increasing. So let's talk about rare genetic disorder in Saudi population. I will start with a newborn screening program, which we established in 2011 in National Guard Health Affairs. And this is part of the National Newborn Screening Program. And as you can see, it is in collaboration with King Salman for Research and Disability and King Faisal Specialty Hospital and Research Center and Ministry of Health. And actually, in this study, 139 hospitals were contributed to this study, and 775,000 newborn were screened. When we screened for 16 genetic disorders, most of them inborn error of metabolism and 2 endocrinopathy. And we found the incidence is 1 in 1,000. As you can see, the disorder, how it is distributed over the region. And we found the commonest is congenital hypothyroidism, followed by congenital adrenal hyperplasia, and propionic acidemia were the 3rd common in Saudi population through this prospective study or a newborn screening study. And we publish this. Also, the other thing indicating the high prevalence of Saudi inborn error of metabolism and genetic disorder in Saudi population. This study which we did is showing that the incidence around 1 in 600. This means that each 1 in 15 individual will carry 1 of genetic disorder. So this is very important to tell you how much these disorders are prevalent in our population. So again, I will emphasize on this comment, inborn error of metabolism or genetic disorder are individually rare, but collectively common. The program will be modeled for precision medicine mainly and personalized genomic. It has a clinical and basic component. It is strengthening the idea of translational research. So I will explain it easily. I saw the patient, or my colleague saw the patient in the clinic. It's an interesting case, non-diagnosed, not resolved. We will take it and adopt it until we find the diagnosis and the novel discovery on this patient. So it's helping the patient, and also it is cost effective. So it is to uncover disease mechanism, to develop a new diagnostic platform, to develop a new therapeutic approach, develop mechanism and strategy to prevent propagation of such disorders. And actually, it is a collaboration between KMARC and university and NGHA hospitals over the kingdom. And also now we're starting collecting cases from outside NGHA hospitals. Actually, this is a central piece of research and development, and it is a mega project in KMARC. All departments participate in this program, not only genomic department. And actually, once we collect the case, we do whole exome or whole genome sequencing, and then we go and do a segregation analysis for other family member, and then functional studies including proteomic, metabolomic, sometime quantitative PCR, Western blot analysis according to the discovered gene or variant. And then sometime we go further to animal model, and finally, we are hoping that we reach therapeutic discovery. So as again, it is a strategical program from KMARC, from all the department together. We have diagnosis, then characterization with biomarkers, and prevention and therapeutic. How we did this program, we start with establishment of whole genome sequencing in our center, and it was really a huge challenge, but with collaboration of the team, we were able to do that. And what I mean by whole exome sequencing, as you know, the gene is formed from promoter and tail, intron, and exons and boundaries, and the most mutation are found in the exons. So we screen all the exon of the whole genome. While whole genome sequencing, it will sequence the whole genome of the human. So we published this, and we found the WIS rate in our population is around 50%, which is higher than Western population, which is around 25 to 30%, and this is because of the high rate of consanguinity. And again, we were one of the first group who published the hit rate of whole genome sequencing, and we found the hit rate of whole genome sequencing was higher by 10% than whole exome sequencing. And to establish this, it is really a collaboration between Bioinformatics department in KMark and IT, and also our genetic and genomic department. I should highlight here, you need high capacity of storage in order to do such a project. So when we did first 100 genome, our data center called us that we really reach a lot of storage of our server, and now we are working with others to able to expand the capacity of the storage. And then we did that, and we established our KMark genome database, and through that database, it helped clinicians to find allele frequency of rare variant among Saudi population, and help researchers to investigate genomic variant rule in combination with other clinical feature for disease development and mechanism. And this database, it will be opportunity also in the future for artificial intelligence and other application for database, as you know. And you can see here we reach now around 2.56 million variant and around 1,500 individual and families. And also we published this recently as the secondary finding in Saudi population. We found that around 8%, which is higher than any other population. So what does this mean? This means when you do whole exome and whole genome sequencing, you will see other diseases other than what related to the presentation or phenotype. The rate of this in Saudi population is 8%. We, through this program, we discover more than 50 novel genes and diseases. As you can see, this is with international collaboration. We discover 2 genes responsible about hyperammonemia. And through collaboration with Professor Al-Quraya and King Faisal Hospital, we discover 33 novel genes in this. And also this disease, we discover it, which also present with a neuroregression, developmental delay, and lactic acidosis. And you can see vanishing white matter in the brain. This disease, after we discover it, as you can see, we make another publication to detail the phenotype. And this is what we did. Once we discover a novel disease or novel gene, we start collecting cases in order to understand that disease. And actually, we published 10 cases from our population and become the international reference for all articles published with this disease. And this is, we call it Escaptogene Defect. Because of the time, I will not go in that, details. But 24 patients has been described so far, and we are the leader of this disease over the world currently. As you can see here, the interesting thing about this disease, the glycine level in the blood is increased and also in the brain. So usually they have an effect in the glycine cleavage, because they have problem with the lipoic acid biosynthesis defect in the sulfur mitochondrial pathway. Again, we were the second group who discovered this disease, which is asparagine synthetase deficiency. And after that, this disease is present with microcephaly, developmental delay, and low asparagine level in blood and CSF. As you can see, the interesting thing about rare diseases, you can see a clear pathway. And this clear pathway would help you to develop the therapeutic approach easily than obesity, cancer, and other kind of diseases like infectious diseases. And this is our publication about this disease. Again, we become reference over the world for this disease. This novel disease also we discover, it's called SLC6A9 defect, which is a GLIT1 encephalopathy. So this is a problem in the transporter of glycine in the brain, and we were the first group who described this disease. They present with high glycine in the blood and in the CSF, but it is different than non-ketotic hyperglycemia, the classical one. Again, from the clinical genetic point of view, we discovered this disease actually 2 years ago, which is present with global developmental delay, intellectual disability, and speech delay. It's called EMC10 gene defect, and we confirm our finding in KMark with quantitative PCR and Western blot analysis, as you can see in the picture. And we were the first group publish this disease. Again, RAP1-GDS syndrome, actually also we are the first group who published this syndrome, where they present with dysmorphic feature, intellectual disability, and speech delay. After our publication, this is the quantitative PCR picture. After our publication, the King Faisal also published this as a part of other genes. And also in Europe, they published 6 novel genes from the Syntogene group. Again, we were the second who discovered this disease, which has a problem in the glycose aminoglycan biosynthesis. And this patient present with a global developmental delay, axial hypotonia, and a problem in the glycose aminoglycan biosynthesis. And also with Dr. Baha Rifai, he's here also hearing us, we discovered this disease, which is a neuromuscular abnormality, infantile spasm, intellectual disability, developmental delay, hypotonia, spastic quadriplegia, and delayed myelination. And interestingly, in this syndrome, we use CRISPR editing in order to prove the pathogenicity. Finally, KIF16B, it's also, we discovered this disease will present with seizure and developmental delay and hypotonia. So in conclusion, genetic and rare diseases are common in Saudi population. The incidence is 4 times higher than Europe and 10 times higher than USA. This is an opportunity for biotechnology companies to invest on these diseases. Kingdom of Saudi Arabia is one of suitable countries to run a clinical trial for rare diseases because we have high number of patients. Logistics should be facilitated in order to shine more in this area. Again, whole exome and whole genome sequencing allow for detection of more novel genes linked to human diseases. The rate of consanguinity in our population is really high, which is 60%, and this really need to more of ultra-rare novel autosomal recessive disorder. We need more larger cohort and follow these diseases clinically with international collaboration in order to reach the detailed phenotyping and diagnose more cases in the future and find therapeutic area. Again, collective and collaborative leadership among scientists and clinicians around the world is essential for gaining better understanding of new genes, pathophysiology and how it relates to human disease, and then moving to identify biomarker with them, and finally, gene therapy. Again, this is not one-man show. This is done with a huge team effort. I would like to thank my lab scientists and the clinicians, Dr. Badr Mzaini, Dr. Yusra Al-Yafi'i, Dr. Abira Tuwajri, Dr. Yazid Al-Hidan, Dr. Muhammad Umair, and our technologist, and also the clinical genetic team, our genetic consultant and coordinator and counselors, and metabolic dietitian, and finally, our pathology department led by Dr. Ahmed Al-Faris and his colleague, and thank you very much.
Overview and incredible work in really identifying these diseases. Am I allowed for questions for Majid? Can you hear me?
Yes, you can. We can hear you.
Okay. I just wanted to ask you, Majid, you showed this incredible data and in identifying rare genetic disorder and obviously all these different mutations. I'm assuming that there are many founder mutations as well. How homogeneous are these diseases in terms of symptomatology and severity? And the reason I'm asking is you've done an incredible work in characterizing all of these, and it seems to me that the kingdom will be a perfect site for many of the clinical trials, mainly because of the characterization that you have in place for these diseases and the ability really to understand the severity of it. So what are you, how homogeneous are, and what are you doing in terms of collaborating more with the companies that have potentially the ability to bring a new medicine for these type of diseases?
Yeah, thank you very much, Paulo, for this excellent question. I think these diseases are mainly neurogenetic disorder with global developmental delay, hypotonia, and some of them are dysmorphic features. Many of them has a biochemical defect. So, for example, asparagine synthetase deficiency has a problem in asparagine synthesis. As we can see, UGDH has a problem in glycosaminoglycan pathway. So I think focusing on that biochemical markers in order to open the horizon for discovery of therapeutic area is really our next step in the future. The companies, as you know, we are not yet reaching the big company in US and in Europe, but we hope that with this work, they will recognize us and come to Kaimark. And this conference actually is one of the way to communicate with the companies and develop our local biotechnology park.
Good. Thank you. Thank you, Majid. Are there any other questions for Dr. Fadel? All right. So should we move to the next speaker, which I think is me? I think, Majid, we did the introduction this morning, so why don't I just go into the, is that okay?
Yes.
Perfect. Sorry, I cannot change the slide because, as you can imagine, the Moderna computer are super protected, as we had a lot of attempt recently of hacking. We are becoming probably a little too famous in the world, and that's it becomes a problem sometimes. So I would like to talk to you today about a little bit. I already gave you an overview on the messenger RNA and why we are making the messenger RNA specific for basically rare genetic disorder. So I'll give you a brief overview on that, and then I would like to introduce you a little bit on the work that we have done on Propionic Acidemia, which is now currently a clinical trial happening in the United States and and eventually all over the world, and in the UK as well. So if we go to the next slide. Next one, please. So I'll go briefly over the messenger RNA, mainly because one of the things that I want to reiterate is that the messenger RNA that we make for rare diseases, which is use a systemic delivery, is very different for the messenger RNA that generally is used for infectious diseases or for other indication. We want our messenger RNA delivered in a systemic manner so it can reach different organs. Unfortunately, the dosing has to be chronic. As we know, unfortunately, messenger RNA is not very stable in time. So we have to do a chronic dosing. That doesn't take away, though, that a chronic dosing, if it's safe and well tolerated, can be certainly happening in patients. And one of the beauty also of the messenger RNA is that it's dose responsive, so that we can really manage the amount of messenger RNA at the expected response from the protein or the enzymatic activity that we expect, so that we can actually be a little bit more precise in terms of dosing and regimen, and also gives an advantage compared to gene therapy in the fact that we can, for example, combine many different messenger RNA at the same time in a single lipid nanoparticle without the limitation of being in a vector or constrained by a vector. And also, then in case of safety, if you have some safety issue, you can certainly stop the therapy and the messenger RNA will basically degrade very fast, as well as the lipid nanoparticle that we have, they're hydrolyzable, so they will disappear in between 8 hours and 24 hours without leaving any residues inside the body. One of the things, obviously, that we've been working on is the immune activation. We know that messenger RNA generally is recognized as a foreign nucleic acid, and therefore is recognized as a virus. But we actually unraveled why the mechanism or recognition of the messenger RNA is triggering an immune response, and we've been able to block that response. So we go to the next slide. And so we have been working quite a bit on messenger RNA in terms of understanding the structure of the messenger RNA, in understanding the stability and the longevity of the messenger RNA, in understanding the immunogenicity of the messenger RNA, and we actually built an incredible algorithm that they take in consideration all these aspects, even when we change the chemistry of the nucleosides, in rebuilding a secondary structure that seems to increment the affinity to ribosome to have a perfect translation. And we can make the messenger RNA also tissue-specific by using microRNAs as a way to dampen the translation of the messenger RNA in certain tissues. Lipid nanoparticle is the vehicle that we use to deliver the messenger RNA. So the lipid nanoparticle that we use, for example, in rare genetic disorder, once again, is very different for the one that was used in the COVID-19 vaccine, mainly because we wanted to be sure that we didn't mount an immune response to the lipid nanoparticle, and more importantly, that the lipid nanoparticle have the ability then to deliver to the cells. And we'll go a little bit in details on the composition of the lipid nanoparticle. And obviously, we do manufacture our messenger RNA, as we know, as we were the first one to basically come with the concept. So we wanted to develop a manufacturing facility that would support not only clinical trial, but eventually a marketed product in the future. And we have a state-of-the-art facility south of Boston, which is only 20 miles from Boston, but that produces right now part of the vaccine and many of the messenger RNA that are in the clinical trial currently. Next slide, please. So for obviously rare genetic disorder, we use IV injection, while for other obviously therapeutic areas, we do either intramuscular, intratumoral, or intracardial. And that's the intracardial injection is actually a collaboration with AstraZeneca, where they're looking at restoring vascularization in ischemic tissues as a result of myocardial infarction. And they have produced incredible data in their phase 2 trial. So we are focusing on IV injection mainly because in rare genetic disorder, we know that we can do IV infusion. And more importantly, we need to target liver and many other organs in certain instances, but with the majority of the liver being the recipient of our lipid nanoparticle. Go to the next slide. And so the lipid nanoparticle obviously are sort of standard, if you will. The only things that we do have differently to the currently marketed lipid nanoparticle is that we have our own proprietary aminolipids. And our proprietary aminolipids are also ionizable, meaning that they have the ability to basically disrupt endosome because of their charges, and also they're hydrolyzable, that they're very fast degraded once they hit the cytosol of the cells when they are internalized as an endosome. So it's a very, we utilize basically the cells and the mechanism and the cytosol to basically degrade our lipid nanoparticles so we don't have any accumulation in the liver of the animals or now in the patients. We use sterols, cholesterols, generally phospholipids, and we also have PEG lipids. And once again, the PEG lipids are proprietary and they've been changed accordingly, and they're also hydrolyzable. And knowing that we have IgG antibody against PEG, having hydrolyzable PEG that they generally are decorating the lipid nanoparticle, the moment that the lipid nanoparticle are injected into the bloodstream, and the blood being aqueous by nature, the PEG lipids are shedded completely from the lipid nanoparticle, leaving basically a circulating lipid nanoparticle naked from the PEG, so there's no interference from the IgG eventually recognizing the PEG and allowing the lipid nanoparticle eventually to opsonize with ApoE and being taken up by the LDL receptor and eventually transduced into the cells. So we get to the next slide, please. So once the lipid nanoparticle release the messenger RNA into the cells, obviously we have a few issues to take care of. One is to be sure that we have enough endosomal escape, that we have enough ribosomal engagement, so that the messenger RNA can translate any type of protein that we can think. We can do secreted, transmembrane, intracellular, intramitochondrial. We really can do it all. And I'll give you an example of propionic acidemia, where we actually deliver 2 different messenger RNA for encoding the 2 major subunits for the PA. So we go to the next slide. And this is just to show you how efficient are our lipid nanoparticle in releasing a messenger RNA into the cells. If you see the red dots in the left panel, those are still lipid nanoparticle encapsulating messenger RNA that haven't been able to release messenger RNA. While if you look at the Moderna LNPs, mostly like 99% of the lipid nanoparticle really release the messenger RNA into the cytosol. And so we have a 25 times better endosomal escape and about 2.5 times a better translational engagement, mainly because when we do codon optimization of our messenger RNA, we look at the secondary structure and we try to build the secondary structure of the messenger RNA that have a very high affinity for ribosome, basically guaranteeing that we have a much better translation compared to the endogenous one. So if you think of, for example, of treating an autosomal dominant disorder, we know that our messenger RNA is going to be translated much better than the endogenous one, and therefore, even if you have a disease with a dominant negative, you can certainly overcome the amount of the dominant negative versus the amount, obviously, of the active messenger RNA and eventually protein. Next slide, please. So we also, as I mentioned initially, we have modified the messenger RNA to avoid the immune system. Messenger RNA generally is recognized as a virus, and so as the ability through the uridine to bind to the toll-like receptor 7 and 8 when they dimerize, as well as to create a second double-stranded RNA once internalizing the cells, and therefore being recognized also by the Rigi complex. And so by changing the uridine, if you go into the next slide, we are capable basically to block completely the immune reactivity of the immune system to the RNA by just using either pseudouridine or methoxyuridine or methyl pseudouridine. They really have the ability to create a steric hindrance to the toll-like receptors so that the uridine doesn't bind anymore to this complex. It does create a much less double-stranded RNA because the uridine is modified. Therefore, we are able to deliver an immune silent and a very highly efficient translatable messenger RNA into the cells. And the messenger RNA that we make is actually the typical messenger RNA you would find in humans, so as a 5' cap, a 5' UTR, a coding sequence, and 3' UTRs, as well as a poly-A tail. So we synthesize the whole things to be sure that it gets translated perfectly by the ribosome. Next slide. And so we see here how basically utilizing our messenger RNA compared to the legacy process, which uses normal uridine, which is in the gray bar. In the blue and green bar, where we use pseudouridine, we completely dumped the activation of B cells in mice, suggesting that our messenger RNA this time is not recognized as a virus anymore. On top of it, obviously, we have a purification process that has allowed us to purify the messenger RNA to a great level, and also that part seems to contribute to a less immune reactivity to the messenger RNA itself, as seen in the gray bar, in the green bar, sorry. Next slide, please. So the last things, obviously, we have all these features for the messenger RNA. We have 5' UTR, which is guaranteed translation initiation fidelity. We can calculate protein per unit time. We have now incredible mechanism of understanding ribosome and codon sitting on the messenger RNA and timing for it in order to create a perfect wild type protein structure. And we can calculate obviously the half-life. And also we can start to make the messenger RNA cell type specific by introducing in the 3' UTR microRNA recognition sites. For example, if we want the spleen not to translate the messenger RNA, we look at the abundancy and the unique microRNA that are in the spleen. And we identify the microRNA recognition site that they're introducing the 3' UTR of the messenger RNA. So the moment that the messenger RNA is transduced into the spleen cells, because of the abundance of a certain microRNA that recognize those particular recognition sites, take the messenger RNA into degradation pathway without the translation of the messenger RNA. So we can make, for example, a liver-specific messenger RNA, avoiding a spleen, kidney, and any other tissues translation because of the microRNA utilization. So as you can see, we always use a very natural processes of making the RNA suitable for human and more importantly, stable in time, and elimination of lipid nanoparticle or endosomal escape by using very simple mechanism of hydrolysis or in these cases, utilization of microRNA for cell specificity. So we don't do anything that is chemically modifying the physiology of the cells. We just use the body to basically make the RNA the way we want it, which I think is an incredible way of thinking about this therapeutic. Next slide, please. So as I said, we actually have engineered LNPs and optimized for the different route of administration. In particular, for rare genetic disorder, they are very much immune silent. And also we have the ability to shed the PEG that potentially could react with the local IgG antibody generated by any type of PEG that we use on a daily basis in creams, toothpaste, etc., and having a perfect delivery into the target organ. We also obviously modify the messenger RNA to avoid the immune system by incorporating modified uridine and eliminating obviously double-stranded RNAs. These allow us to be a little bit more precise in delivery and more importantly in dosing and regimen, because at that point we know exactly that the messenger RNA will not be destroyed or immune compromised, but will be delivered at a certain amount that will give a certain amount of protein. And obviously, we also codon optimize the messenger RNAs and UTRs in order to favor translatability, which allow us to really treat every type of disease in autosomal dominant disorder, because the way our messenger RNA is recognized as a much higher affinity compared to the endogenous one. And we can make a cell type specific, as I just mentioned to you, using the advantage of utilizing microRNA. So in the next slide, given that we actually have the ability to create these messenger RNA that they're immune silent and particularly well translatable, I want to give you an example of the propionic acidemia, which is now mRNA 3927, which is currently in the clinic, and we just terminated the first cohort, and we got the approval from FDA to move to cohort 2 because of safety and tolerability. And so we are very happy with that. And we are starting to see great signs of the messenger RNA therapy in rare diseases as well. If we go to the next slide, I know that Majid or Dr. Alfadel just spoke about propionic acidemia as being one of the high prevalent diseases in the kingdom. And that's why I wanted to talk to you about what we are doing in particular. And we also have a methylmalonic acidemia program, which is just started in the UK in the clinic. And I know that these are highly prevalent diseases in the kingdom. That's why I wanted to give you an overview on what we are doing based on the animal studies and how we got to the clinic. So in the case of propionic acidemia, there are two major subunits that are involved in terms of mutation. There's PCCA and PCCB, and there are mutations in both subunits. So in theory, if you use a single messenger RNA, what happens is that you might correct only one defect in one subunit. What we actually thought about it, since these two subunits come together to create an important complex to process propionyl-CoA into methylmalonyl-CoA and eventually to be part of the Krebs cycle, was actually to create a drug that would contain both messenger RNA for the PCCA and PCCB, so that we don't discriminate anymore of the different type of mutations for these patients. So this is an advantage of the messenger RNA because we can combine as many messenger RNA as we want and to tailor the therapy based on the type of mutations that you have. And if you have more subunits mutated, we don't need to discriminate anymore for the single patients with a single mutation, but we can treat every patient with that disease because we are basically reconstituting the whole complex without having to focus exclusively on one mutations or the other. So if you go to the next slide, obviously, a little bit of an overview on the propionic acidemia is an organic acidemia. As I mentioned, it's a defect in the propionyl carboxylase alpha and beta, and when the two comes together, they create this hexadodecamer complex that is very stable. But once you have a mutation either in the alpha or in the beta subunits, this complex basically doesn't form in many instances, or if you have only one subunit, there's the other subunit that is missing, and both subunits start to actually disappear. And so that's why we decided to combine two different messenger RNA that would allow us not to deal with potential lack of an endogenous level of the counterpart subunits that was not mutated. So it's a pediatric disease, mainly because these patients have difficulties in passing the 6 years of age due to metabolic decompensation, frequent metabolic decompensation. And right now, the only therapy that is approved besides the diet, which is very brutal for these patients because has to be deprived of many protein and amino acid. So the only care for the moment is liver transplantation and diet that not necessarily resolve the issues of this disease. And so in the next slide, please. So what we have done, basically, we, in a healthy, obviously, propionic acidemia patient, the hexadodecamer, the two subunits will come together, will create this big complex, and process the propionyl-CoA into methylmalonyl-CoA. When we have a patient that has a mutation in one subunit or the others, basically, propionyl-CoA cannot be processed to methylmalonyl-CoA, and it starts to produce metabolites like hydroxypropionate, 2-methyl citrate, and ammonia, which are very toxic for the body. So what we have done, next slide, please. we actually have combined the two messenger RNA, and we wanted to test the ratio of the two messenger RNA subunits in order to understand what was the best input of one subunit versus the other, or if we needed both subunits at the same level. As you can see in the first panel, we are just testing a single subunit in a fibroblast from PCCA patients or PCCB patients. And we see that once we replace with both, we actually get a greater activity rather than the single subunit, which is the lower light blue bar, while the big blue bar is the combination of the two. If we look actually at the ratio of the two subunits that we need to give to cells, either in PCCA patients or in PCCB patients, we see that basically 1-to-1 ratio is the one that works the best. And so these actually gave us the opportunity to then start to look at those dependencies increase in PCCA activity. And we see that obviously we see a dose-dependent increase in activity once we give the two subunits in 1-to-1 ratio in a dose-dependent manner. And that's why we decided to make this combination as a 1-to-1 ratio of a messenger RNA encoding for PCCA and encoding for PCCB as well. If we go to the next slide. What we obviously look always is the localization, and in this case, the mitochondria is the organelle where the PC complex has the activity, and so you can see in the last right panel that's the colocalization of the PCCA subunit and PCCB subunit within the mitochondria, merging with obviously the staining for the mitochondria, and so therefore. We decided that we could move into animal and do some experiments in animal to see the efficacy of the combination of the two messenger RNA. Next slide, please. We did studies, and this actually was an inspiration in my conversation with Dr. Majid Al-Fadel when they started the carbaglue studies in human, suggesting that there was at least an impact in dropping the level of ammonia in these patients. And so we did the same in mice using carbaglue or carglumic acid in as a single treatment. And also we wanted to use our messenger RNA to see how comparable in dropping the level of plasma ammonia the messenger RNA was to carglumic acid. As you can see, both the blue bar and the orange bar, where orange is carglumic acid and blue is the messenger RNA combination, those have the ability to drop the level of ammonia similar to the hypomorphic mouse, the heterozygous, which is actually non-affected. And so you can see that the messenger RNA has the ability to drop the level of ammonia equally to carglumic acid. The difference, though, is that with the messenger RNA, not only we dropped the level of ammonia, but we were able to see actually dropping level of 2-methyl citrate, C3C2 propionate ratio, and a 3-hydroxypropionate, both in plasma and in liver, which actually is a great features to have because we are not only dropping the level of ammonia, but we are also dropping all the metabolites that seems to have an impact on the disease. So we go to the next slide. We also wanted to do a multi-dose studies to show that the consistency in repeat dosing. And so you can see that we went up to 12 weeks, dosing every 3 weeks. And every time we dose in a dose-dependent manner, we actually are able to drop the level of a plasma biomarker. In this case is on the far left is plasma 2-methyl citrate. In the center is 3-hydroxypropionate, and then on the right is the C3C2 ratio. And you can see that we drop the level and then they come back, and then when we retreat, the level drops again. And more importantly, even after the 5th dose, we still see this drop, suggesting that, in fact, we don't have anti-drug antibody formations in this case, and we can continue dosing without any immunogenicity issues. The things that I think is important to note that once you express a protein in the liver, I think the liver start to work towards the tolerization, and there's been proven in many different papers now, and even in some of the gene therapy studies, by targeting the liver, even in a knockout model, you start to tolerize for that particular protein, and this is why we can do multiple dosing without seeing actually anti-drug antibodies dampening the effect on the enzymatic activity of the PC complex, which I think is a great feature of that. Next slide, please. So obviously, not only we look at multi-dose studies, we're obviously looking for a consistent response, particularly in 2-methylcitrate, but what we notice also that the mouse model that we use has the ability to develop in time a little bit of a cardiomyopathy. And we saw actually that with the treatment, after the 5th dose, we started to see a reduction in the heart rate as well, suggesting that probably we did have an impact on cardiomyopathy as well in time. So this was actually great news. And then when we went to FDA to file an IND, they were very pleased with the data, and they were very pleased with our toxicology package with our lipid nanoparticle. And so they gave us the okay to enter a phase 1/2 trial, which is a multiple ascending dose directly into patients, which is unprecedented. And they gave us the opportunity to start the first cohort between 6 years of age and above. And then the second cohort, we can open up to 1 year of age and above. As they understand the safety of the system of this platform, they gave us the opportunity really to treat very young patients from the very beginning. So this is good news. As I mentioned, we just completed the first cohort of these phase 1/2 studies, and we got the okay to proceed with cohort number 2 because of safety and tolerability. And so now we are starting recruiting for cohort 2 for propionic acidemia patients. And we just started also the clinical trial for methylmalonic acidemia, which is actually all the preclinical data are published, and you can see them online at this point. So in the last slides, I think, I just wanted to conclude saying that we have successfully identified a lead candidate, which is a combination of the 2 messenger RNA encoding for PCC and PCCB. The duration of effect is about 21 days in mice, and we have a significant improvement in ammonia level comparable to carglumic acid, in serum biomarker, and more importantly in heart rate. And we know that these patients generally develop cardiomyopathy, and that could be also one of the causes of death. And more importantly, we retain the therapeutic efficacy throughout repeated dosing, suggesting that probably we do tolerize by expressing in the liver the protein, and we don't see today the anti-drug antibodies. So this was great news. So with this, I want to thank all the people at Moderna. This is just a fraction of them. This was a picture taken before COVID, and now if you look at the picture post-COVID, that room is not enough anymore to contain all the people, because the growth for COVID has been absolutely important in order to provide the vaccine to all over the world, but also to concentrate now on this rare genetic disorder that seems to have, a messenger RNA seems to have an impact as well. So keep your finger crossed, and potentially we'll have a therapy for propionic and methylmalonic acidemia soon. Thank you very much for your attention, and I'll be happy to answer any question. Okay, if there are no questions, okay, we can actually go to the next presentation. The next presentation is given by Dr. Hisham Feniri from Northeastern University, and he's going to talk to us about magnetically guided rosette nanotubules siRNA delivered for cancer gene silencing. Dr. Feniri, please.
Dr. Martini, thank you for your introduction. If I can have the first slide. None of my clicker is working. Is it on? Good. Okay, good. So on this first slide, what you see is a set of molecules that are entirely synthetic. There's no equivalent to these molecules in nature. And they have the ability to self-assemble spontaneously into superstructures that I will show you in the next slides. So on this slide, you see some of the applications that we have explored with these materials. In the center of the slide are 5 nanotubular structures that result from the spontaneous self-assembly of this synthetic moss. And around those structures, we have a variety of applications, such as drug encapsulation and delivery. We use them for tissue engineering and screening for medical devices and for a compatibilization of implant devices with human tissue. We have also functionalized them with a variety of nanoparticles, such as gold, platinum, palladium, copper, silver. And we have used some of those nanoparticles and nanotube hybrids as antimicrobials, and we have shown that they have very effective in antibiotic-resistant bacteria. We have also used them to deliver microRNAs to reverse inflammation in patients, not patients, but these are actually models of patients suffering from atherosclerosis. And we have also functionalized them with certain peptides to deliver them on the right panel, bottom right panel, to deliver them to human lung adenocarcinoma cells and induce programmed cell death or apoptosis. So these molecules are shown here. They are inspired from the natural molecules, so we're deviating dramatically from the particles that were described by Dr. Martini for delivery of messenger RNA and small interference RNA. These are a different architecture and different structures. They are derived from nature, but they're not natural. So we're combining a guanine and cytosine into a new base called the GC base that we can synthetically modify into a variety of derivatives. And on the next slide, what you will see is this structure has the ability to self-assemble into a super macrocycle that you see here. It's a ring of 6 molecules maintained by 18 hydrogen bonds with nanometric dimensions. And then this structure spontaneously self-organizes into a tubular structure. We can control the dimensions of the system, length, diameter, functionality on the surface, functionality inside the nanotube, and stability of the system, the thermal as well as the physical and chemical stability of the system. So I'm not going to get into the details, but just to show you what they look like, this is an electromicrograph that shows the self-assembled nanotubular structure. In this case, they're extremely long. We haven't controlled the length in this case. We want them actually to grow as long as possible. And this is just a close-up view of those structures. They are 3.5 nanometers in diameter. And so they express a certain number of chemicals on their surface, and we have complete control on the nature of those chemicals on their surface. So on the next slide, you see the 5 families of nanotubes that I mentioned in the second slide. And they are, as you can see here, this is the GC, this is the twin-base GC, this is the tricyclic equivalent of the first one. We call XK1. This is one that is functionalized with a group that points towards the interior or the lumen of the channel, therefore modifying the physical and chemical properties of the nanotube, and therefore the ability to capture molecules that are not compatible with physiological media and can deliver them, as I will show you in the next few slides. And the one on the right, YK1, is the largest one. We made that one actually for a different type of application in molecular electronics. Okay, on this slide, what you see is an encapsulation of a molecule inside these nanotubes. And those plots that you see next to the nanotubes, those are the potassium mean force, which is basically the association, the energy of association of this molecule inside the channel. And you can see as this channel as these peaks become higher, it means that the molecule have more and more difficulties to move along the main axis of the channel. And so therefore, by changing the nature of a nanotube, we can control the height of these peaks and therefore control the rate and the rate of release and the stability of the molecules inside those channels. So we have looked at the drug data bank, and the drug bank against these 4 nanotubes, 7,000 drugs or so, and we have a very beautiful scattering between 750, actually between 500 and 1,000, showing that there is some selectivity for certain molecules. It is more obvious on the next slide. On this slide, you can see, for example, in the case of dexamethasone, which, as you know, is an anti-cancer drug and also an anti-inflammatory. It's used actually in the case of COVID patients. This molecule has different affinities for the 4 nanotubes, and it varies between -7 kilocalories to -16 kilocalories, therefore allowing a fairly nice control over the encapsulation energy and release of the molecule. Not only that, we can actually tailor these molecules with radioactive elements for SPECT imaging, with fluorine 18 or 19 for PET imaging, or with fluorescent probes for FMT imaging. So they can be not only delivery agent, but they can also be traced using existing techniques for imaging. So it is, in a sense, a theranostic tool for delivery, for diagnostic imaging and drug delivery. On the next slide, what you see is the same series of nanotubes. Here on the left is a nanotube functional with the lysine amino acid, therefore the surface is very positive, charged positively. On the second nanotube with the red peptides, that's an RGD peptide that we know binds very strongly to integrin receptors. And on the third and fourth nanotube to the right, those nanotubes are a hybrid of the first 2. And therefore that gives us some leverage on controlling the density of functional bioactive molecules on the surface of these nanotubes. And we have used that trick to, decide the nanotube that I mentioned to you. And using this methodology, the clicker doesn't advance as quickly as I want. So these nanotubes functionalized with these peptides can actually bind to the overexpressed receptors, integrin receptors on human lung adenocarcinoma cells, and trigger the MAP kinase pathway and lead to programmed cell death of this human lung cancer cells. Now, the next application draws actually from all this knowledge, and we have looked at this nanotube in a different way. We have thought that by functionalizing them with a varying degrees of charges, of positive charges, we can control the charge density on their surface, and therefore the affinity for negatively charged molecules such as siRNA and messenger RNA. And we have tried these systems with different functionalities on the surface to deliver siRNA. So the molecules, this is a very schematic description of the process. The molecules self-assemble into rings, the rings self-assemble into tubes. The tubes bind to the siRNA on the surface and form particles. Those particles show a net positive charge. They are recognized on the surface of the cell, endocytosed, and released inside the cell. And then the siRNA expresses its activity inside the cell. So what happens when you mix siRNA with these nanotubes? On the left panel, so this is the schematic. On the left panel, here is a transmission electron micrograph of the nanotubes without siRNA, and you can see that they have that typical rod-shaped structures. But on the right side, with siRNA, you see particles on the micrograph. And in fact, when you zoom in on these structures, what you see is that there are very beautiful hexagonal structures of this nanotube viewed from the top. So each one of these white dot corresponds to a nanotube viewed from the top. And the dark area between the nanotube is where the siRNA is trapped. So these are the structures that are bioactive. And so when we look at how this siRNA and RNT bind to each other, we did a gel shift assay, looked at the amount of nanotubes that could bind to an siRNA, and we found actually there is a very nice correlation between the length of the nanotube and the length of the RNA. And the RNA can grow almost indefinitely. However, in these cases, the growth of the nanotube is controlled by the length of the RNA itself. And you can see here that from the zeta potential, we can determine the charge, the net charge of the nanotubes alone, K1 and K3. And here, the negatively charged siRNA. It has a negative zeta potential. The complex is positive with K1 and even more positive with K3 because K3 have more charges. So this is what we draw from this. Obviously, this system is also very stable to enzymes. So the siRNA is fully protected from oncogenic enzymes. When you look at this slide, we looked at the cytotoxicity of the cells, of this complex, and we found that the RNAs are actually non-toxic up to 100 micromolar, which exceeds by far the amount needed for the therapeutic effect that we are seeking. On the next slide, we're looking at a variety of cells. We have actually screened a battery of 60 different cells, and we have found that the nanotube with siRNA cargo enters the cell within minutes with an efficacy between 98 and 99%. So it's an extremely efficient delivery system for siRNA. And then we looked at the gene knockdown of these structures. And in this case, we have looked at, we compared this to lipofectamine, which is considered to be the gold standard for in vitro systems. You can see that K1 and lipofectamine are essentially the same against these two cells. And here, this is just additional examples for siRNA that sign a STAT3 protein, the expression of STAT3 protein. So on the next slide, what you will see is an attempt at silencing KRAS. KRAS gene is expressed in 90% of pancreatic cells. And here what we can see is on the left side is a delivery of all star cell death siRNA. You can see that the effectiveness of the lipo is slightly higher than K1 and K3. And when we do that for the KRAS expression in cancer cells, what we see is actually the efficacy is essentially similar to lipofectamine. The advantage, however, in this case is that the concentrations we use are significantly lower. And secondly, our material is not toxic. In addition to that, we have taken advantage of the open channel of these nanotubes, and we loaded it with gemcitabine, which is known to be one of the effective drugs against pancreatic cancer. And we found actually that the efficacy of the drug is amplified by a factor of 3. So this leads me to the last part of this particular section of my presentation. And that leaves me with 1 minute 30. So I don't have time to get into the detail of this process, but these are magnetically guided RNA delivery system. We have magnetic nanoparticles that we can coat on the surface. On their surface, we can coat nanotube, and using layer by layer deposition methodology, we can actually layer nanotubes and then siRNA, multiple layers. We can guide these nanoparticles using a static field, so we can aggregate them so that they can respond to magnetic field. We can drive them to the site of action, and then we can use a dynamic field to separate the particles from the aggregate, and then those particles can easily enter the tumor. So that's where we are currently. Before I end, I would like just to tell you about this is a slide that describes the magnetic delivery. And before I end, I would like just to tell you about the foundation that I represent as well. It's Masir Foundation is a nonprofit organization in Morocco, and this foundation has been working for a number of years on promoting science, research, innovation in Morocco. But we have collaboration across the world, established in 2007, and has grown into a very respected research foundation in Morocco, focusing on microelectronics, biotechnology, and material science. And we have, in the area of biotechnology, which is relevant to this conference, we focus on medical biotechnology and green biotech, especially developing diagnostic tools for a variety of disease. So it was actually the foundation that created the first COVID test in Morocco, and it is now used across the country. And I'll also mention that we have tremendous amount of collaborations since 2007, where now this slide goes to 2020. And we are expanding this network, and we're open. This foundation is a nonprofit foundation and is looking for expanding its reach and collaborating in a variety of fields across the world. And with that, I'd like to thank you for your attention, and I'm happy to take any questions you may have.
Thanks very much, Dr. Fernieri. That was very interesting. And I have a couple of questions. Otherwise, I want to see first if anyone has any question. Otherwise, I'll ask my question. I'll start to ask a question then. You show nicely the conjugation of these nanotubes with silencing RNA, and you mentioned also potentially messenger RNA. It looks like from your schematic that the silencing RNA is sort of attached to the nanotubes on the outside. And when you try messenger RNA, do you see the same? Because I would imagine that probably you will get degradation of messenger RNA faster than a silencing RNA.
We have worked with the short messenger RNAs up to about 300, and we're looking into much larger structures for now. Up to 300, they're perfectly stable, just like siRNAs.
Okay. Great. The other question is, once I saw that obviously once you bind to the receptor, you mentioned it gets internalized with a great efficiency. What's happened to the nanotubes at that point? What is the degradation pathway? What is the accumulation if it happens?
So we use a very, very small amount of these nanotubes. So we're talking about nano or sub-nanomolar concentrations. Okay. These molecules, as I showed you in the previous slide, they are derived from natural molecules. So it's peptides, and the bases are mimics of the natural bases. We are assuming that they are degraded in the same metabolic pathways that degrade peptides and nucleic acids. We haven't looked actually in detail because the question never, well, obviously this question we have thought about it, but it did not rise to the level of investigation yet because all the animal studies we have done didn't show any toxicity.
Okay, great. Thank you very much. I appreciate it. If there aren't any questions, we can move to the next speaker. Yes? Okay. It's my pleasure to introduce Dr. Madhuri Hegde. She's from PerkinElmer, and she is Senior VP and CSO at PerkinElmer Genomics, and she's giving a talk about Beyond the Exome, whole genome sequences for rare diseases diagnostic. Dr. Hegde, please. Welcome. You're on mute.
Hello.
Thank you.
Thank you, Dr. Martini, and I'm glad to be here, and thank you for the opportunity to speak today. So what I'm going to talk about is we're going to switch gears a little bit into diagnostics of rare diseases. And essentially what we are going to discuss is how we are moving towards whole genome sequencing from the exome. And what I want to start with today is to talk a little bit about my own background and a little bit about PerkinElmer and how this lab really is operating on a global basis. So as we talk about PerkinElmer Genomics,
We lost Dr. Madhuri.
So what we're going to talk about today is the whole genome sequencing for rare disease diagnostics. But if we look at a little bit about PerkinElmer for a few minutes, because it's really important to understand where PerkinElmer Genomics sits today. My own background is in academia. I am a clinical molecular geneticist, and I have worked in diagnostic testing for almost 25 years. PerkinElmer Genomics exists within the ecosystem of PerkinElmer. And the reason I mentioned this today for rare diseases is that we just heard a talk on the RNAi therapeutic side of it, that we are very rapidly moving towards therapeutics. And what we can do now is bring all of the diagnostics, applied genomics, life sciences, analytical tools together to do diagnostic testing and identify these individuals or patients who can benefit from a variety of different therapeutic approaches. So just as a quick background of PerkinElmer Genomics, the foundation is in newborn screening. As we all know that newborn screening is done across the globe. About 150 million babies are born every year, out of which 55 million babies on a regular basis actually receive newborn screening in some form or the other. What we did at PerkinElmer Genomics was build on that foundation and expand into genomics with a full service clinical laboratory, and now a near-term expansion is going to be in omics. The idea really being that we want to do a continuum of care model going beyond traditional genetic screening and testing. So what do we actually mean by that? What we are looking at here on this slide is the mother, the infant, and the adult family. And what we are now able to do today with the advances in genomics and omics is do sequencing, biochemistry, and now even with NIPT, using a complete omics capabilities, and that has been our goal from day one. So with that said, just key facts about PerkinElmer Genomics. We have 9 global locations in India, China, Malaysia, Sweden, and the US. As I mentioned, I am from academia, so I myself have published extensively, but together we have almost 300 plus peer-reviewed publications. And we continue to expand our capabilities based on the most recent technological advances and the scientific knowledge that we gain from these advances. So with that introduction, let's jump into whole genome sequencing. You know, the title of this talk really gives you that perspective of that we are trying to go beyond the genome or beyond the exome. And what does that mean? So if we just today look at the milestones we have achieved in genomic testing, you know, for those of you who remember doing Sanger sequencing using radioactivity followed by the fluorescent labeling, and then now the most recent advances going towards the next generation sequencing technologies, and we're going to continue making some rapid advances into nanopore technology. In our own laboratory, we do sequencing using the NovaSeq. We have 10 NovaSeqs in our lab, with the idea being that are we going towards whole genome sequencing? So let's spend a couple of minutes looking at how does whole genome sequencing actually work. Before that, let's look at panels and exomes. Essentially, when we are doing panel and exome sequencing, what we are doing is selecting the regions of the genome to amplify, sequence, to identify the possible variants that could be causing the disease. So here on this slide, what you see is in our laboratory, we use a sure select system from Agilent. We have over 300 panels and the whole exome, which we can sequence using this sure select library. Now, what are the drawbacks of exomes and panels and a target selection method? So in my experience, I've tried everything from PCR-based amplification selection to hybridization-based. And there are plenty of publications now which have shown that hybridization-based methodologies are superior, especially in rare disease diagnosis, to the PCR-based amplification methods. But we still have that PCR step, which can create a bias or an allele dropout and thereby introducing errors. So what we are trying to do here is, can we go straight from our DNA to sequencing itself? And that's where whole genome sequencing comes into play. What we are essentially doing in whole genome sequencing is taking the naive DNA, ignoring all these middle steps, shredding that DNA into tiny pieces, and then sequencing it through a massively parallel sequencing, or NGS. Now, just one disclosure here, that whole genome sequencing does have a step of PCR, but it's minimal. It's called tagmentation, and Illumina has a nice video on their website of how this tagmentation actually works. So how do we bring whole genome sequencing to the clinic? On this slide, it really shows you how it was important for our laboratory, the PerkinElmer Genomics lab, to be within the PerkinElmer ecosystem, and how we are using this instrumentation in our own lab. The first thing which we did was, the reason I showed you newborn screening is that newborn screening is done on dried blood spot cards. So today, we can do whole genome sequencing on a dried blood spot card. So we have validated whole genome sequencing on dried blood spot card, saliva, blood, and also tissue types. Now the extraction is done on a Chemagic 360. Chemagic 360 is a very robust instrument for DNA extraction, but recently in the COVID times, it has been widely used for RNA extraction as well. Then DNA quantification is done using the Victor Nevo, followed by library preparation on a cyclone liquid handler, again QCing on the lab chip GX, and then followed by sequencing on a NovaSeq. All this exists within our laboratory, but we also attempted to write our own analysis software, which is called ODIN. ODIN really stands for Ordered Data Interpretation Network. And what ODIN really does, it grabs variant data from the globe from our own labs and helps in data interpretation. As we all know that there are, though we are more than 99% similar, there is that minor variation which a clinical geneticist has to look at and differentiate between disease causing or common variation in that population. So let's compare the sequencing options here. Panels, exomes, and genomes. The panels and the exomes target a region, so they give you 100x coverage, a higher depth of coverage. Today, the genome is sequenced at 30x or 40x. But because you're not doing target selection, a 40x genome is comparable to a 100x exome or a panel, or at times better than an exome and a panel. Because the genome now allows us to detect copy number variation, at times some pseudogenes, and I'll talk about that, and also repeat regions. Incidental findings need to be addressed across the board from panels, exomes, and genomes. And I'll give you just one simple example, which is the Gaucher disease gene GBA, which is implicated for Gaucher disease and Parkinson's disease as well. Now, if we do a direct comparison of the depth of coverage, as I just mentioned previously, that a whole exome at 100x is directly comparable to a whole genome at 40x, and a 40x genome is at times better than a 100x exome. And the reason for that is the uniformity of the sequencing itself. What you see in the top panel is exome sequencing with the troughs and the peaks, whereas in genome, you get that uniformity of the sequencing itself. The next thing to look at is the coverage of clinically significant regions. And this slide really says it all, that from the ACMG selected genes to the SGMD database genes to the RefSeq genes, whole genome sequencing gives you better and a more uniform coverage. A question that gets asked very often when I give this presentations is, does genome sequencing give tremendous amount of data and does it make it very difficult to interpret? I think let's just compare directly to the number of variants in an exome versus a genome, exonic and total variants. On the lower two panels circled in the green box, you see the exome to genome comparison there. If you look at just at the exonic variants, the variant number is quite comparable, where you see this number of genes here and the number of variants. The genome definitely gives you more variants, but you can limit what you see because the deep intronic regions can be investigated in a more tiered fashion. Now, GC-rich regions, as I mentioned, the coverage is more uniform. So what you see here on this slide is on the left-hand side, you see where the whole exome coverage drops off and the uniformity of the coverage from whole genome sequencing across the x-axis, which shows you increase in the GC content itself. On the right-hand side is a single exon comparison of an exon, which is extremely GC-rich, 77%. And you can see that the whole genome can cover that exon versus the exome. Actually, you get a dropout of that exon. You cannot sequence it using whole exome sequencing. So beyond the exome, the genome really has that ability to capture those variants which we cannot capture using exome sequencing. There are lessons learned. We can identify those deep intronic variants and then tag them in the exome and make the exome powerful as well. So basically what you're doing is investigating the entire genomic sequence of the individual to make sure that you have looked at the gene in its full completeness, but not just the exon and the covering or the flanking intron exon boundaries. Now, let's look at some couple of cases here. The first case is an intronic variant case because I've just talked a lot about having that extra sequence which is a deep intronic sequence, which can become very important. And I'm going to show you a very simple example. You would expect this to be a simple molecular confirmatory step post biochemical analysis. So this is a 5-year-old male with a suspected diagnosis of MPS II, commonly known as Hunter syndrome. Biochemical results showed reduced iduronate sulfatase levels, and Sanger sequencing and MLPA could not identify the sequence variant which was associated with this child. The family was really frustrated and approached us for whole genome sequencing to identify the variant. And what we found was a variant in intron 6 at -72 position, which was causative of this particular condition, the Hunter syndrome, in this child. Subsequently, functional analysis was done, and we were able to move this variant of unknown significance to pathogenic in this particular child. So now this really shows you the power of whole genome sequencing. Let's go to the next step, is detection of tandem repeats. What I've shown on this slide is again a very simple example of the PABPN1 gene, which causes OPMD. And what you can see is on the top panel is the whole exome results, and the bottom is whole genome. And the genome can actually resolve ambiguous repeat sizes detected in exome sequencing. We have done extensive validation of repeat regions, and what I wanted to show you is how those reads actually look. So on the top panel, you see the whole exome data, and the bottom, you see the whole genome data. What you can see is that region where the PABPN1 repeat region sits is not well covered by exome, but is completely covered by whole genome sequencing. So we have done extensive validation of the repeat disorders by whole genome sequencing, and we are doing about 40 disorders now. We can report them. What we are doing is we can rule out, but we cannot rule in. And what I mean by that, repeat disorders which are in the smaller range, where the repeats are about 10 to 40, we can easily detect them, even the expanded repeat, by whole genome sequencing. When the repeat size goes extremely high in 2000 and 3000, we cannot detect that today by whole genome sequencing. But if we see heterozygosity or heterozygous repeats in the normal range, we have then essentially ruled out that disorder. An example will be Fragile X in a male or even a female at times with skewed X inactivation, or the smaller ranges of the normal repeat range in myotonic dystrophy, the DMPK1, where if we see homozygosity for the normal repeat, then a southern blot would be needed for confirmatory testing. This has been very, very useful for those cases which are coming to our lab with specific clinical conditions such as ataxia or a myopathy. Now, the next level of whole genome sequencing is the ability to detect SMN1 and SMN2 copy numbers. And we are today routinely reporting SMN1 and SMN2 copy number from whole genome sequencing data. What you see here on this slide is a publication from 2017, which essentially shows you the region of investigation here and the SNPs that are actually investigated or looked at to distinguish between SMN1 and SMN2. As you all know that it is not very easy to do that because it's a single nucleotide change in exon 7 that really separates the two genes and one deep intronic change. And we have been able to extensively validate that and now are routinely reporting the SMN1, SMN2 copy number, which is important for therapeutic purposes, as we know. And the last one is exon level detection. With a genome and exome sequencing, essentially what we do, and most labs do, is take that data, create virtual probes to detect the copy number variation. But because of the uniformity of the data that you get from whole genome sequencing, it makes it more powerful. What do I mean by that? Can I demonstrate that? So let's look at an example, is utilizing the power of whole genome sequencing for single exon detection in the presence of a larger detection on the other allele. So this is a 6-year-old male with intellectual disability, abnormal hands and legs, speech problem, and there was no family history given to us. What you see here on this slide is a really power of whole genome sequencing where you can detect a deletion within a deletion on both alleles. We've been very fortunate that we have been able to extensively partner with advocacy groups. What you see here is Decode Duchenne through the Parent Project Muscular Dystrophy, because we sequence the entire Duchenne gene. We also have a project called the Lantern Project with Genzyme, which has been very successful for muscular dystrophies. But really the key is, how do we separate ourselves and make this whole genome sequencing technology more amenable to the patients? Is ability to work with any type of sample, the extensive breadth of the menu itself from newborn genome or newborn screening to adult testing, and then the global laboratory network, which is essential to understand the genomic variation globally. So I mentioned previously, we have labs globally. You can see on this slide, really the labs which are present globally, and we congregate all this data into our OD database for data analysis, for improved and tailored variant interpretation. So with that, I'm going to stop and take any questions. Thank you.
Thank you so much, Dr. Agde. Great presentation, great talk. Let's see if anyone has a question. Otherwise, I obviously have a question for you. It's very interesting. No, I'll start at this point. It's amazing. I'm always in awe with the progress in technology and how it goes so fast. So my question is actually this. It's impressive how now we can sequence whole genome basically from different patients, and I see that you participate in newborn screening. But how fast can we actually do a full sequence? And the reason is that I'm asking is because if we think of some of the liver metabolic disorder where we have basically only a few days before these children start to develop metabolic decompensation, and before we understand, for example, if it's an organic acidemia or a urea cycle disorder, because we see ammonia, but we don't differentiate between the two. So how fast can you produce the data so that we can, in fact, one day, once therapy will be available for infants and newborn, produce this data that allow us then to be very precise with the type of therapy that we give to these patients?
That's a great question. So today you can, from the time you receive the sample, you can produce the data in about 3 days. What we can do is we can, it depends on how quickly the sample comes to the lab, obviously. We've been doing stat sequencing with a turnaround time of about 7 to 10 days. It really is including, from the time the sample comes to the lab, the DNA extraction piece of it too. But on the sequencer, you can generate data in 3 days. Now, you raised a very good question is, for the newborns especially, which are in the, NICU, who need that data so quickly so that could be some more action oriented. And what we are now able to do is take that data and marry it with the biochemical data so that if there is a disorder especially when there is a disorder that is treatable, you really want to get in there very fast before the clinical presentation has advanced too much. And we are able to do that now and combine the data with newborn screening data as well. And that is why, right, blood spot cards also become very important because you don't have to go back to the baby. We can just take the blood spot and sequence it.
Perfect. That's phenomenal. Thank you so much. I think it's incredible that we can do this. I remember 20 years ago, we were still running gels, counting base by base. And now we have 3 days genome sequencing. So it's phenomenal. Congratulations. Thank you.
Thank you.
I don't think there are any questions, so I'll move to the last speaker of this session, and then we'll have an open panel for discussion with all my colleagues. So the last talk is given by Dr. Majed Algoribi from KMark, and it's talking about empowering infectious disease surveillance using whole genome sequences. Dr. Majed, please.
Thank you, Paolo, for this introduction. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Today, I'll take you away from the whole genome sequencing of human to whole genome sequencing of infectious disease, especially in the pandemic, the COVID pandemic situations, how the infectious disease surveillance program using the power of whole genome sequencing play an important role to understand the evolution and the transmission dynamic of COVID all over the world. If you allow me, I will start my presentation, with this quote by the great microbiologist Carroll, who contributed in the Tree of Life. And he said in the 1998 that genome sequencing has come of age and genomic will become central to microbiology's future. As we are witnessing nowadays that the whole genome sequencing, especially with COVID pandemic, play an important role to understand the virus, the movement of the virus, and the evolution of the virus. Infectious disease surveillance is a cornerstone of public health. Without infectious disease surveillance, then we'll be able to identify the emerging and re-emerging pathogens that hit the world. As I said, by having infectious disease surveillance, that will help the community and the public health to understand the threat of infectious disease. The traditional surveillance program was using the public health data and the clinical data and diagnostic data. But recently, I think starting from 2003 up to 2010, there is a revolution of whole genome sequencing, next generation sequencing technology that's come to the science where has been used for infectious diseases. As I said, COVID is a point, become a turning point of genomic history, where whole genome sequencing of COVID never been done before in any other pathogens in the history. Especially for the global surveillance of SARS-CoV-2, we have witnesses a cooperative approach of all countries where they have implemented whole genome sequencing surveillance to identify the whole genome sequencing of the virus all over the world since the start of the pandemic. There is a lot of initiatives has been contributed to the surveillance of SARS-CoV over the last year and a half. And there is a huge database has been created using all the generated data by every country to see the dynamic transmissions of the virus all over the time since the pandemic started. One of the most famous and important database is the GIS-AID initiatives, where all the scientists and research centers and public health agency have deposited whole genome sequencing of the virus since the start of the pandemic. Other tools of the bioinformatics and database has captures these data and put it in a platform where these data has been interpreted using bioinformatic tools to identify the variants and the transmission of the virus. Can you imagine that only in 1 year and a half, almost 1 year and a half, almost 3.5 million whole genome sequencing of the virus has been generated by all over the countries, all the globe. This has never been done for any microbial organism or even human genome. This is a very huge data, is enabling us to understand the evolution of the virus in a very short time. As you can see here in the left tables, that the total genome sequencing has been done by each country. As you can see here, the United States has generated almost 1 million whole genome sequencing of the virus, coming after that UK. But to compare that and to see the contribution of the whole genome sequencing, we should do a bit of statistic to see the sequencing per 1,000 cases. As you can see here, some of the country, they have almost sequenced most of the cases that is reported in their country, including Ireland, Iceland, and New Zealand, and Denmark. That really helps these countries to improve their infection control precautions to contain the virus, and also to understand and to determine the variant that's circulating in these countries. Also, by having this huge amount of whole genome sequencing data, we were able to do identify the classifications of the variants. As you can see here in the chart, the first classification of the virus is the variant of high consequences. This is, fortunately, that's never been determined. That is really more severe and make the patients having really severe infections that allowed to stay in the hospital for a long time. The second one, which is the most worrying nowadays, is the variant of concern. We have around 4 variants of concern nowadays circulating worldwide. And also, there is many variant of interest that has been monitored by the Public Health Agency to monitor these variants to make sure that it is not becoming a variant of concern. Also, many countries have identified variants under monitoring. This is still under monitoring, and there is many of them in the database. This is the most famous variants has been detected over the last year. Alpha, which is belong to the B117 lineages, and this is first detected in UK. And also beta, which is similar to having similar mutations to the alpha, has been detected in South Africa. In KMark, we have recognized the importance of whole genome sequencing surveillance of infectious disease early on, starting from 2015. We have been working on this to establish a state-of-art whole genome sequencing facility in KMark to be able to tackle any issue in infectious disease. The vision of this surveillance program is to halt the dissemination and reduce the threat of infectious disease in Saudi Arabia and the regions. And our objective is to conduct a larger-scale national-wide surveillance to track routes of disseminations and monitor the trend of emerging, re-emerging pathogens over the time in Saudi Arabia and the region. If you allow me, I will take you through our journey of developing this surveillance program, where we were concerning about building the capacity, transfer the technology and knowledge to our center to be a kind of a leading center of whole genome sequencing in the region. We're starting by building capacity. We were focusing to establish the microbial genomic lab to bring the most advanced sequencing platform in our lab. That will allow us to sequence pathogens that never been sequenced in Saudi Arabia to generate cutting-edge research in the region. All that is coming together to serve as a surveillance program to monitor the dissemination of high-risk pathogen in Saudi Arabia. And also we were focusing here to build the human capitals and train scientists and technologists in our center. Also, we are serving as a reference lab to most of the National Guard hospitals, where we serve this lab when there is a difficult diagnostic of a certain infectious disease in these hospitals. This is including a manual detections of unknown infectious disease, unknown pathogens, using advanced molecular techniques. In 2018, we started this surveillance program by building a strategy of infectious disease in KMark, where we have the planning phase, where we start by designing the experiment and making networking with all the National Guard hospital in Saudi Arabia to be able to create a comprehensive surveillance program by designing a random screening as well as design screening for a certain pathogen that is become a high priority in the region. Also, at the same time, starting with 2019, we've been able to bring the whole genome sequencing platform in KMark, where we're able to sequence these pathogens. I will give more details about the whole genome sequencing platform that we have in KMark. Nowadays, we are in the analysis phase and development phase, where all these whole genome sequencing generated in our lab are being used to develop rapid diagnostic tests, or also to contribute in the vaccine development. We have in our department Illumina platform. Because, as you know, the genome of the microbe is very small comparing to the human. And Illumina, they have different sequencing platforms, starting from MySeq, which is a small throughput sequencing platform. This is kind of suitable for a small lab who is working to sequence minimum around maximum 100 pathogens per month. We're starting by the small-scale proof of concept to make sure that we have developed the pipeline from the sequencing up to the bioinformatic analysis, where we can interpret that data and understand the evolution of any pathogens we are sequencing. The only problem with this approach is required a manual approach to do a library preparations, but that can be done, can be avoided by developing automated library preparation. We moved. We have this facility in our lab as a small scale when we need it, but also we try to have a high throughput sequencing platform with a collaboration with the MGI. We have in our center an automated automated machines for library preparations, and also an automated machine for nucleic acid extractions. All this, when we go back to Illumina, here we can sequence around from 25 to 30 bacterial pathogens. This is a small scale, and also for viruses, we can sequence up to 100 virus. This is depend on the size of the genome of the pathogen. When we compare it to MGI, this machine can sequence around 300 bacteria in 2 days. And that will help us to move from a small to high throughput scale, and also it's important to associate that with an automated library preparation. Just going back to the surveillance program, we have 5 national hospitals in the region, in Riyadh, Dammam, Al-Hassa, Al Madina, and Jeddah. All these hospitals are sending a sample on a monthly basis, started from early 2018. And we have designed a selection criteria for this pathogen to be sent in every month. And then after that, we collect all the clinical data associated with this pathogen, just to kind of to have a platform where we can have a selection criteria for whole genome sequencing. Not all the isolates are sending from this hospital are sequenced, but we do a selection criteria for whole genome sequencing, just to have a designed sequencing surveillance. There is 3 core components of the infectious disease surveillance system. The first one is the surveillance site, which is each hospitals, and each hospital they has a clinical microbiology lab and also infection control department. We are coordinating with this department where the clinical microbiology lab they send us the isolates and all the diagnostic data, and also we communicate with infection control to collect any epidemiological data that associated with each sample. By this, we have created a database for pathogen biobank. All this is taken to be in one platform. The second component of the surveillance system is the AMR NGHA Coordinating Center, which is KMARC, where we collect all these isolates by a team of networking and communication, where they communicate with these hospitals. But also we have a data management and quality control, where all the isolates and the data go through a quality control pipeline to make sure that we are not missing any important information. The third component is the lab, where these isolates, when they arrive to KMARC, we make 3 copies. 2 copies go to different biobank for security of these samples. And also, once we review all the data, we put these samples in a pipeline. Some of them, they just go for the pathogen biobank, and some of them go for further molecular investigations. Then we will confirm that these isolates is meeting the selection criteria for whole genome sequencing. Then we'll take these for whole genome sequencing stage. I'm not going to go in details, but this is the workflow and the pipeline of microbial genomic. We do a manual phenotypic genotypic detections, and also we do a whole genome sequencing ending to the bioinformatic analysis. We have managed to develop an in-house bioinformatic pipeline for antimicrobial resistance bacteria. We have a specific pipeline for each pathogen. For example, we have several pathogens that we collected over the last 3 years. Each pathogen have its own database, own pipeline software that will enable us to generate enough data to understand the evolution of the virus, of the bacteria. In addition, we started in 2019 by sequencing MERS-CoV, which is the virus that hit Saudi Arabia in 2012. And because the number of the number of cases was very low at that time, we managed to collect around 50 whole genome, 50 isolate of MERS, and we worked to develop the viral whole genome sequencing pipeline during 2019. By the end of 2019, we managed to sequence 50 isolates, and that will enable us to be ready to sequence any type of viruses that we gain in good experience over that sequencing. Before we go into the SARS-CoV whole genome sequencing, our experience in KMark, let's start with antimicrobial resistance. This is the main surveillance system that we built. We used the most priority pathogens that has been identified as a high-risk pathogen in clinic: E. coli, Pseudomonas, Klebsiella, and Acinetobacter. We collected more than 20,000 pathogens over the last 3 years. All these isolates being through the pipeline for a selection criteria for whole genome sequencing. This is a number of publications that we have generated over the last 3 years, starting with the message that we have sent to this community to introduce whole genome sequencing in our region. With collaboration with Public Health England, as I did my postdoc in Public Health England in London, I tried to transfer their experience in a national surveillance program to Saudi Arabia. And we put this position article in the corner in the left there as a guideline, a roadmap for whole genome sequencing surveillance program. Also, we sequenced several pathogens, including E. coli, Klebsiella, that's showing a pan-resistance. Pathogen are resistant to all the antibiotic available. All this experience is cumulative to build all the protocols for an effective surveillance program. On the other side, we were trying to cover a little bit in a parallel to clinical pathogens. We were trying to look for animal and food microbiology, including Salmonella and Brucella. This is just an example of our analysis. We usually end up with genome analysis of high-risk pathogens that are harboring genomic elements, for example, like this circular plasmid, which is harboring the resistant genes that enabling the bacteria to emerge and become more resistant. This analysis is a downstream analysis. Just we look for the genes and the mobile element, including the plasmid, the insertion sequencing of these pathogens to understand the evolution of the antimicrobial resistance in the region. For each country, usually there is a type of resistant genes that are circulating all over the country, and we are trying to take the surveillance program to the national level and also not only to the national, regional, and hospital. We call it local surveillance to understand if each of these plasmids are circulating in one city or circulating in one hospital or even circulating in one ward as an outbreak in a hospital. This is another example of one of the first KBC genes that are detected for the first time in Saudi Arabia. I think it's been existed in Saudi Arabia for some time, but because there is a lack of whole genome sequencing surveillance in the region, we were able to pick up the first case in Saudi Arabia. Taking all this experience together, we have been invited by the WHO to meet, to visit Geneva for the first meeting of the GLASS whole genome-based MR surveillance committee. This was held early in 2020 before the COVID, but that enabled us to develop a technical note where we can use our experience and other country experience to be together in this technical note to help other countries to implement whole genome sequencing surveillance in their countries. Going back to the COVID, MERS, and SARS viruses, as I said, we have gained a great experience dealing with MERS from human and camels. We have sequenced a good number of isolates comparing to the cases reported in Saudi Arabia. And also, by March 2020, when we have the first patients in National Guard, we were able to have the RNA of the virus in just 1 day. And when we got the RNA, we've been able to sequence the virus only in 3 days. All over the time since the starting of the pandemic, we've been sequencing in a designed screening. We are not sequencing every case that we have in our hospital, but we've been sequencing from time to time. By the end of 2020, we have observed one of the interesting mutations that has appeared in our sequencing, which is the 501T. This is similar mutations that has been detected in the UK and South Africa variant, and then we focused to look for this variant more further. Then after, in around February 2020, we sequenced a huge number, 300 isolates from 1 month. We found that 60% of these isolates contain this significant set of mutations that never been reported worldwide. Then we kept our sequencing facility going to see if that is a real variant or variant of interest or variant of concern. We collaborated with the Infection Control Hospital in Riyadh, and we have collected all the clinical data of the patient who been infected by this variant. We found that only around 6 patients from around 150 has been admitted to the ICU. Most of the cases were community-acquired infections, and that was a relief for us because this is a not high-risk pathogens, not a variant of concern, but variant of interest that we need to keep watch all over the time to make sure that is not really causing a severe infection. Our experience here helped us to see where we can track these variant all over the time to see the source of the variant. We have done a global phylogenetic analysis. We took a prospective whole genome sequencing from all over the globe to compare our isolates with the other variant reported. As you can see here in the middle, there is a clade in a red color having the same mutations that come into our interest, the 501T. But when we look for that, we found this is a cluster of isolate that has been sequenced and isolated from monk in the United States early in the middle of 2020. But as you can see here is the line where is the present and absence of this variant. In the left side of the slide, you can see the red, there is a clade that is having a significant set of mutations not existed in the other isolates. And in the bottom of the screen, this is a zoom in to that tree. As you can see, these isolates mostly detected in Saudi Arabia started from November 2020 until we have a peak in February 2021. As you can see in the left side, these isolates Saudi Arabia has reported the most number of these variants comparing to other countries. We worked closely with the NextStrain. NextStrain is a platform for phylogenetic analysis and clade analysis of each of SARS-CoV-2 viruses. We have built a national platform, Saudi Arabia platform, to compare our variants that we are sequencing all over the pandemic. As you can see here, there is a clade with the alpha variant and the gamma variant and the beta variant. And the clade in the yellow color, it's the Saudi variant that we have detected, which is fortunately is not a high-risk variant. I think our experience over the last 4 years become a kind of a proof of concept to establish a whole genome-based surveillance program where we really saw the important and the major role of whole genome sequencing to identify the evolution of the emerging and re-emerging pathogens, where we need always a real-time data on occurrence of high-risk infectious agents through a standardization method. We need a standardization method that is really meet with the international standardization protocol, just to make sure that when we share our data and compare our data with the global data, then that will be compatible. Also, there is an urgent need to enhance sample and data collection for the national level, and also we need to develop more molecular investigations methods across all the compartments of human, animal, and health. Detections of related infectious agents in the time and the locations through phenotypic and the genotypic. We need to combine all the data that generate in the phenotypic and the genotypic profiling. Also, we have to think about the possibility to observe trend in clone variant as we have experienced in SARS-CoV, and also the ability to rapidly compare between all the type of data using a common language. I think one of the most important messages that I would like to send tonight is the national capacity building. We need more whole genome-based surveillance system in the country, not only in KMARC, but also we need collaborations and consortium of whole genome sequencing surveillance where we can all share data and then create a comprehensive pictures of the epidemiology of infectious disease in Saudi Arabia. Without monitoring and surveillance program, we are blind to the infectious disease issues. Developing an effective surveillance program aims of the reference labs in every region and country, each serve as the hub and national network and contributing to the international network. Each performing essential testing, including both phenotypic and genotypics testing, each following international standards. And also, one of the most important elements for this type of surveillance is sharing data. No real-time surveillance without no real-time data sharing. That means we have to create a common platform where we can deposit all the data for the national surveillance system, just to be able to compare that data and then come up with a clear pictures of the pandemic, of any issue in pathogens. By the end of this presentation, I would like to thank the IDRD team in KMARC who is contributing to establish the AMR surveillance program, and also all the hospital, National Guard Hospital in Saudi Arabia. Thank you so much.
Thank you, Dr. Al-Ghoribi. This is phenomenal. Congratulations on the incredible work. You're truly contributing to the safety and the health of the world by doing this work. And I'm glad that you're publishing, and more importantly, you're planning to share and contribute and collaborate with the international organization. This is very important, as we have seen what happened with COVID-19 that created a pandemic. So congratulations and thank you for doing that work. It's very important. I don't know if we have time for another questions. Unfortunately, we need to probably move to the panel, and we got 10 minutes before the Master of Ceremony will close the meeting today. So I would like to pass it on to my colleague, Dr. Majid, if he has an initial remark for the panel. Otherwise, I can start as well.
Thank you, Paolo. You can start, no problem.
Okay, I just, first of all, I want to thank everyone for participating in this session. It's been incredible. We have seen an incredible presentation today, but more importantly, what I see that there is a vision from the kingdom to really not only contribute with the rest of the world in the safety with obviously this incredible presentation and also the ability to really maybe start to build something with the vision of helping the patients in the kingdom. So this meeting for me was an incredible experience and humbling, as a matter of fact, in understanding the vision that this country has in moving forward, but not only in rare genetic diseases, but infectious diseases, and etc. So I just I wanted to congratulate everyone for this, and more importantly, I would like to start maybe a conversation on what is the next step for KMark and eventually for the kingdom to really start to tackle down what are the key issues and the key implementation that need to happen in order to bring potentially more medicine and more ability to run clinical trials within the kingdom and eventually to contribute to the human health.
Thank you, Paolo, for this excellent exposition. I think in KMark and all other institutions in Saudi Arabia and research centers, we didn't have deficiency in science or in brains. I think what is the issue that we need here is to facilitate mainly the logistic. So we need the international companies to present more here and develop their infrastructure here so we could exchange the information, utilize the rich, resources that the country have. And I think this summit is the initial step toward this endeavor. I am sure with the 2030 vision, this kind of collaboration and effective communication between industry partners and KMark and other research centers will be more and more in the near future as reflected by the summit.
Great. I think it's very important as we see now new technology coming to life. It was great to see Dr. Faduri presentation as well, new possibilities for targeting tissues and different diseases. So there's quite a bit of growth and development in the technology. Also, obviously I presented about messenger RNA. But right now it seems like the next step is really to be able to access all the information that you guys have been collecting with great publications and more important with incredible work that has been done even in rare genetic disorder, what you presented in terms of understanding mutations, diseases and the characterization of the diseases is an incredible work that not many people can actually do. And you have obviously the ability to contribute incredibly to the patient population by obviously creating these databases of patients that you guys have, and it's just incredible. So I would like to hear from other colleagues if they have any remarks or questions.
I just have a question, Pablo, if you allow me.
Yeah.
I want to ask Dr. Maduri regarding whole genome sequencing. I know Birken-Elmer sequence a lot from Arab population. Are you willing to share the data that you sequence with our research center and other institutions in Saudi Arabia? Because, you know, this data is very important in order to reach the precision medicine in order to have our reference genome ready. We have a lot of variant of uncertain significant because actually we lack the reference genome. And I think if we have collaboration with an international company like Bergen-Elmer and Syntogene and other companies who enter the market in the Middle East, I think we will reach our reference genome easily.
We will be very happy to collaborate and share the data. It is not an issue at all. In fact, we do a lot of sequencing from your region on a daily basis. We are doing a tremendous number of whole genome sequencing cases. I myself report at least 20 to 30 whole genome sequencing cases from your region on a weekly basis. So we do have a tremendous amount of data, and you are absolutely right. This is the only way to resolve the variants of unknown significance. Because otherwise, if we don't collaborate and share that data, these variants sit as variants of unknown significance. There's another initiative I have started, and I would welcome the collaboration there, is that many variants of unknown significance, as Dr. Martini mentioned, is liver diseases or any pediatric disease can be moved to pathogenic if the physician has made the diagnosis and started some sort of a treatment and has received a response. We are collaborating with a lot of pharmaceutical companies to enable them to get into clinical trials into various regions across the globe. So we'll be very happy to collaborate and share data.
Thank you. The other question I have regarding the storage, when we did our whole genome sequencing in Kmark, we faced these challenges of storage. Are you keeping the BAM file and other file, or you just save the VCF file? And tell me about your experience on storage, how you overcome this.
So we store all the FASTQ files. We do not discard any FASTQ files. And every 6 months, I can tell you, we routinely get asked by physicians to reanalyze the data. And we find new genes and new variants that can be moved from genes of unknown significance to a gene causative of disease. So we store all the FASTQ files and routinely move them into our Odin software for reanalysis and reinterpretation.
Thank you, excellent. The other question to Paulo, you know that many now technology is running regarding the therapeutic genomic, including CRISPR, including microRNA, including messenger RNA. Why your company choose messenger RNA? Why you didn't go to CRISPR and these technologies?
So we are not excluding these, right? Right now, when Moderna started, they wanted to focus on messenger RNA, and they put a lot of effort in understanding messenger RNA and to make it become basically the best therapeutic so far in the clinic. I just don't want to brag about it, but as you have seen many of the data recently, that they've shown the superiority of the Moderna vaccine compared to other, and mainly due to the fact that the company has invested a lot of money and a lot of people understanding messenger RNA, lipid nanoparticle, how to create the perfect, for example, a spike protein for a vaccine as an antigen, et cetera, et cetera. So when you invest so much in understanding the type of platform and therapeutic that you have, you focus initially on really socializing that and then making it become a real therapeutic. It's not excluded that in the future, in fact, and we are already looking at other technologies that involve nucleic acid, because after all, nucleic acid is our expertise, which will potentially include the use of aptamers, silencing RNA in combination with messenger RNA, or a CRISPR technology using messenger RNA as a way to deliver the protein, the Cas9 or whatever. So we are thinking about that, but for the moment we were focusing exclusively on messenger RNA because we wanted to become really the leader in the world in understanding what messenger RNA is, how we can make it suitable for human, and what type of delivery we need in order to target many different diseases. We still have a long way to go. The CNS, for example, is an organ that is still tough to address, right? And lipid nanoparticles seems not to be, for the moment, the way of delivering. So it's always interesting to understand also from Dr. Feniri, for example, a different way of targeting a delivery system, using a delivery system. So for us, messenger RNA right now is the future. It doesn't mean that we won't explore any other nucleic acid, because as we build expertise in that and we are becoming the leader in the world, we want to be sure that we create the perfect medicine, as perfect as it can be for these patients, right? And so you won't be surprised in the future if you see messenger RNA combined with different nucleic acid, because of the complexity of also of the disease that we are going to face, right? Right now we are sort of exploring the low-hanging fruit, infectious diseases, vaccine, monogenic disorder. But in the future, we will need to probably target different diseases that are more complex, and different technologies are needed in order to be able to be a little bit more specific in the therapeutic intervention.
The other question, the prevalence of rare diseases in Saudi Arabia is really high. I mean, it's, as we mentioned in our lecture, it's around 4 times higher than other population. However, we see the clinical trials conducted in United States and in Europe. What do you expect, or why, what is the driver for you to go to Saudi Arabia and do the clinical trial there? And what is the challenges right now you are facing that limiting you from thinking about this solution?
So, and I'm generally very direct and honest on these. The challenges right now is that obviously phase 1 trials has been run in other countries because there's experience in running phase 1 trials in those countries. But this doesn't take away that, as far as I understand it, you guys are actually, in fact, getting ready, and also already got it ready for phase 1 trials. So my collaboration that I've been discussing with Dr. Houdi is, in fact, to come to the kingdom and to start to run the first phase 1 through the kingdom, mainly because I would like to see this happening. You have an incredible characterization of your patients. A clinical trial in your country will be much easier to run because of the understanding that you have on the patient population, the stratification of the patients that you have, making calls on the patients that are eligible for the clinical trial will be much easier in the kingdom, in fact, because of the characterization that you have on the mutation and the metabolites and everything. What I've seen in the past few years when I came and visit, there was an incredible effort to characterize these patients that for a phase 1 clinical trial is absolutely important and paramount to be sure to select the right patients for the trial. So I would love to come. One of the things that was initially posing us was the ability to actually do a phase 1 trial, which I believe now you are getting ready for it. And that's why we are discussing a potential collaboration, because I think it will be important, in fact, to come to the kingdom because of the homogeneity of the studies that we could run and the response to the drug that we could have because of the data that you have on the patients. So as you developed more the phase 1 trial capabilities at Kmark, I think you will attract more not only Moderna, but many other companies that will feel at ease to be able to do such work in a country that doesn't have an history for phase 1, but has a great history for phase 2 and 3, right? So, and when you test a new drug, is always tough, right? Because there's always the potential of toxicity, tolerability, etc. Right now, we are starting to have our own initial data from the trial in the US and the UK on safety and tolerability. So as we feel more at ease in proposing our drug to many different countries, I think the Kingdom is the next best place to do it. So I'm planning to do that.
Sound great.
Yeah, and I think we are discussing, in fact, a collaboration, and I'm hoping that within a few months we'll be able to finalize it. And there's quite a bit of work involved in that, and I'm so proud to be able to have initiated that, because I think it is the future.
Yeah, that's great. One of the most important elements that we should develop in the national level, that, as Dr. Majid Al-Fadl mentions, the data and the storage of the data. Not only the storage, but we are really need to have a high performance computing that allow to do a high throughput sequencing analysis, which need a power of computational power that enable to do a sequencing. Fortunately for bacterial pathogen, bacterial genomic, it's easy. It's a little bit doable. But when it comes to the human genome, and then I think me and Dr. Majid always, we are fighting for to run a job in our high performance computing, and we have usually waiting for the whole genome sequencing of the human to finish, and then we are waiting to put our jobs. I think one of the excellent example I have experience in England. They have built a cloud high performance computing, a bioinformatics where people able to submit jobs and use that for a high throughput analysis. Maybe in Saudi Arabia, I think one of the initiatives that should be planned is to have high performance computing that serve the academics, research, and clinics.
Yes, I agree totally.
Good. I think we are running a lot out of time, but you guys tell me, otherwise we can close this session and give the time to the Master of Ceremony to close the meeting. And so I would like to thank everyone. Thank you first of all, thank you for inviting me. It has been an incredible experience. And it's been always truly informative to be able to share incredible presentation with my colleague today in the session. And thank you for an incredible meeting. This was amazing. And thank you for the organizer to invite me as well.
Thank you.
Absolutely. Thank you, Dr. Paolo, and your esteemed panelists for a great session. Ladies and gentlemen, we have reached the end of the Riyadh Global Medical Biotechnology Summit 2021. I would like to express my appreciation to all the participants for taking time out of their busy schedule to attend the summit virtually for 3 days. And thank you all to the speakers who joined us from around the world and shared with us their insightful views and rich knowledge. And it was my pleasure to be the Master of Ceremonies for the Riyadh Global Medical Biotechnology Summit 2021 and work with our amazing speakers locally, regionally, and internationally. And now for the closing remarks, I want to welcome with great pleasure Dr. Abdel Ali Al-Hawdi, Head Strategy and Business Development, Managing Director, Medical Biotechnology Park KSA, and the Vice Chairman of the Riyadh Global Medical Biotechnology Summit 2021. The stage is yours, Doctor.
Thank you. Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Good evening, good afternoon, and good morning to you all, wherever you are following us in the Riyadh Global Medical Biotechnology Summit 2021. As just been said, we are now reaching the end of this summit, but it's not the end of it. It is actually just the beginning of a long journey for this great country, where medical biotechnology will be a mean to position the country as a gateway for medical biotechnology, actually a global gateway for medical biotechnology. Now, Let me tell you how we get to this wonderful, fascinating expression that I just put there. If you look at the program of the summit from day one until today, 9 sessions, if you look at them, it was not actually a random or random selections of topic somehow related to medical biotechnology. It was actually a clear attempt to take all of you, including, of course, the great people of this country, the region, and around the globe, on what does it take to go from great science, that is really the basis, to an economic impact. So we started with the high-level considerations. What are the elements of a biotechnology setup ecosystem, incubators, biotechnology parks, science and technology parks, you name it. Some of them are sharing facilities, some of them are sharing platforms, some of them are sharing management structures, funding facilities and support. Diving now into really how those companies are successful. What do they need to do? Shall they call on CROs, CDMOs? How do they really develop their product? How do they manufacture? What does it take? Now we're going backward. Backward because really we start the right way. The right way for us is that we start by developing a strong foundation for solid science. Without that great science, we cannot move forward. Now it's time to start translating that science. And here I would like to quote my dear colleague and friend, Ron DePino, when he said that great publications will not cure patients. But the translation of those great publications into products, into devices, into drugs, into vaccines, that will cure patients. And that's exactly what we're attempting to do. Whether it's through Kmark or other institutions in the country, that is the way forward. Now, from that consideration, we give you highlights through a number of distinguished global guests from biotechnology companies, pharmaceutical companies, academic elite institutions from around the globe, but the science, genetics, genomics, gene editing, cellular therapy, stem cells, gene therapy, vaccine research, vaccine development, all of it. So in reality now, if you put all those nice sessions together, you will feel that is a complete story, complete journey that we are all attempting to go through. A few minutes before I came to the stage, my colleague just told me that we had about 175,000 viewers of this summit from over 150 countries. That is quite impressive while we are at the beginning of this journey. And hopefully, next time when we meet, we will tell you more about where are we, what we have been able to achieve, and hopefully what we can do together with local partnership and our global partners. Without any further delay, I would like to call my great colleague, Dr. Ahmed Laskar, the Executive Director of King Abdullah International Medical Research Center and the Chairman of the Riyadh Global Summit for Medical Biotechnology 2021. Dr. Ahmed.
Thank you, Dr. Abdul Ali, Hawdi, and what a wonderful summit. I'm really thrilled and impressed, not only with the attendees and the intensity of attendance and the viewing, but with the program and the whole content of these last 3 days. It's been wonderful 3 days that have been rich with science, exchange of expertise, collaboration, signing MOUs, and most importantly, the will and the enthusiasm that we have seen and witnessed and felt throughout these 3 days. Although we have most of the attendees are online and at a distance, however, we see the enthusiasm and and the intensity again to learn more about this biotechnology. As you said, Dr. Abdul Ali, we are not only restricted to science, but we ought to move forward again to translate this science and those publications into tangible products that will impact not only the health of the individuals and the communities, but also the global economy. And we see this, fueled with the, in the Kingdom of Saudi Arabia with a vision, the Vision 2030, that wanted to have alternative to the oil production, and that would be science and innovation. I would like to thank His Royal Highness Crown Prince Mohammed bin Salman for his patronage and for leading this vision, which this ought to be part of it. And also I would like to thank the Minister, His Royal Highness, the Minister of the National Guard, Abdullah bin Bandar bin Abdul Aziz, again for his attendance of the opening ceremony and for his follow-up of the agenda and the program for this summit. I would like to thank the attendees, participants, and the speakers, and all of you guys, and those organizers. There are so many people that ought to be thanked, again, for putting this all together. I wouldn't say goodbye, but we ought to see you again in another meeting and another conference, and hopefully, most importantly, to translate what's been discussed and what's been signed into reality. So we need to see it on ground, and we need to work hard, again, to translate all of these discussions into tangible products and with our collaborators. So without further ado, again, thank you very much, everybody, for your attendance and for staying late tonight here in Saudi Arabia. However, we shall see you in another conference and another meeting very soon. Thank you.
الآن وقت الصورة الجماعية اللي أجلناها إلى آخر الحفل. لو تتفضلوا جميعكم على المسرح.